If you are studying or want to study microbiology, then this video is for you. Assalamu alaikum guys, I am Afsana. You are cordially welcome to my channel. In today's video lecture, I'll be talking about the history of microbiology. Microbes or microorganisms are minute living things that are usually unable to be viewed with the naked eye, such as bacteria, fungi, protozoa, algae, and viruses, etc. The microbes have coexisted with humans from the beginning of civilization, providing both beneficial and detrimental roles to human life. And microbiology is the study of these microbes. To know about the origin of these tiny little things, we need to explore the history of microbiology. A brief history of microbiology. The history of microbiology can be subdivided into 4 different parts. The first one is the discovery era, second one is the transition era, third one is the golden era, and the fourth one is the modern era. Here in this picture, we can see some of the famous faces who have contributed a lot building the history of microbiology. Now let's discuss about the discovery era. During 348 to 322, Aristotle and others believed that living organisms could develop from non-living materials, which is now known as the spontaneous generation or abiogenesis. Spontaneous generation or abiogenesis is an early hypothesis about the origin of life, which states that living organisms arose from non-living substances, such as maggots from meat. This concept was so compelling that it persisted until late into the 19th century. However, a strong opponent of spontaneous generation, the Italian physician Francesco Redi set out in 1668, even before Van Leeuwenhoek's discovery of microscopic life, to demonstrate that maggots did not arise spontaneously from decaying meat. What he did was he filled 2 jars with decaying meat. When the meat jar is left open to the air, flies lay eggs on the meat. As a result, the hatches transform into maggots. Proponents of spontaneous generation believe that rotting meat gives rise to the maggots. But when Redi closed the jar and sealed it with a cork and a gas, the flies couldn't get in. No maggots appeared in the meat, and Redi concluded that the maggots came from the flies and not from the meat. In the year of 1665, Robert Hooke was the first to observe the smallest unit of tissues, which he called cells. Here in this picture, we can clearly see the cells. In the year of 1676, Antony van Leeuwenhoek was the first person to observe and accurately describe microorganisms. That time it was bacteria and protozoa, which he called animalcules, which also means little animals. The microscopic observation of different kinds of cells are illustrated here. As he was the first person to produce precise and correct descriptions of microbes, he is considered as the father of microbiology. Leeuwenhoek is also considered to be the father of bacteriology and protozoology, or protistology. The term microbe was first used by Sidyat in 1878. Now the transition era. Let me first start with John Needham's story. John Needham, during 1713 to 1781, probably the greatest supporter of the theory of spontaneous generation, proposed that small organisms, animalcules, appear spontaneously in his mutton gravy. He covered the flask with a cork like Redi did, and he also warmed some flasks. However, microbes appear in the mutton broth. Lazzaro Spallanzani, during 1729 to 1799, refutes Needham's experiment. He boiled beef broth for a long time and also removed air from the flasks and sealed them. After incubation, no growth was observed in these flasks. He also showed that the heated nutrients could still grow animalcules if exposed to air. Thus, Spallanzani disproved the doctrine of spontaneous generation. Sulch, 1815 to 1873, and Theodor Schwann, 1810 to 1888, proved that air was the source of microbes. George Schroeder and Theodor van Dasch were the first to introduce the idea of using cotton plugs for plugging microbial culture tubes in 1854. Now let's move into the golden era. During 1857 to 1914, about 50 years. The golden age of microbiology began with the work of Louis Pasteur and Robert Koch. During this period, we see the real beginning of microbiology as a discipline of biology. The controversy over spontaneous generation. The issue was still unsolved till 1858, until the German scientist Rudolf Virchow questioned spontaneous generation with the concept of biogenesis. Biogenesis is a theory that asserts that living things can only be produced by another living thing and not by a non-living thing. But Rudolf Virchow couldn't offer any scientific proof, so arguments about spontaneous generation continued until 1861. However, that issue was finally resolved by the French scientist Louis Pasteur. Louis Pasteur, 1861. By a series of experiments, Pasteur demonstrated that microorganisms exist in the air and can contaminate sterile solutions, but the air itself doesn't create microbes. What he did was, he boiled broth in a flask with a straight neck and left it exposed to air. Organisms grew. When he did the same thing with his goose-necked flask, nothing grew. The S shape of this second flask trapped dust particles from the air, preventing them from reaching the broth. By showing that he could allow air to get into the flask but not the particles in the air, Pasteur proved that it was the organisms in the dust that were growing in the broth. Thus, he disproved the spontaneous generation theory. That's why he is called the father of modern microbiology. He also found that the fermentation of fruits and grains resulting in alcohol was caused by microbes, and also determined that bacteria were responsible for spoiling the wine during fermentation. Pasteur in 1862 even suggested that mild heating at 62.8 degrees Celsius, approximately 145 degrees Fahrenheit, for 30 minutes rather than boiling was enough to destroy the undesirable organisms without ruining the taste of the product. This process was called pasteurization. His work led to the development of the germ theory of disease. Louis Pasteur is also known as the father of bacteriology. The term microbiology was given by Louis Pasteur. John Tyndall, during 1820 to 1893. He discovered highly resistant bacterial structure, later known as endospore. Prolonged boiling or intermittent heating was necessary to kill these spores, to make the infusion completely sterilized, a process known as Tyndallization. Robert Koch in 1876. He was the first one who proved the germ theory of disease. It states that certain diseases are caused by the invasion of the body by microorganisms. Pasteur put forward this theory, but was unable to prove it. The first proof that bacteria actually cause disease came from Robert Koch in 1876. He also depicted some guidelines used to prove that a disease is caused by a particular microorganism, which are known as Koch's postulates. I have described the germ theory and Koch's postulates broadly in a different video, so you can watch them as well. Van-Alchemie's Hayes, 1850 to 1934, one of Koch's assistants, first proposed the use of agar in culture media because of its higher melting 96 degrees Celsius and solidifying 40 to 45 degrees Celsius points than gelatin, and was not attacked by most bacteria. Richard Petrie developed the Petri dish, a container used for solid culture media, in 1887. Thus, the contribution of Robert Koch, Van Hayes, and Richard Petrie made possible the isolation of pure cultures of microorganisms and directly stimulated progress in all areas of microbiology. 1865. Joseph Lister, father of antiseptic surgery. He introduced antiseptics in surgery by spraying phenol, which is also known as carbolic acid, on surgical instruments, wounds, and dressings. He reduced surgical mortality due to bacterial infection considerably. Edward Jenner was the first to prevent smallpox during the year of 1749 to 1823. He also discovered the technique of vaccination. Paul Ehrlich, 1910, introduced the concept of chemotherapy by developing an arsenic compound called salvacen to treat syphilis. In 1928, Alexander Fleming discovered the first antibiotic, penicillin. Lastly, let's move into the modern era. There are four main scientific advances that form the basis of modern microbiology. These are: first one, the invention of the hybridization probe. Radio-labeled hybridization probes were developed in the 1960s by Joseph Gall and Marie-Laure Pardoux. Number 2. Discovery of the polymerase chain reaction, or PCR. In 1983, Carey Mullis developed polymerase chain reaction (PCR), which enables a target stretch of DNA to be copied thousands or millions of times. Third one, the observation that the microbial species signature can be read in the ribosomal genes and that it can also be read in the proteins is the last one. I have also added some of the contributions of many microbiologists of the modern era for giving you some knowledge about them as well. For example, in 1901, von Behring won the Nobel Prize for discovering depth antitox. In 1902, Ronald Roche, and in 1905, Robert Koch disclosed the organisms causing malaria and TB respectively and won the Nobel Prize. In 1908, Metchneka found out about phagocytosis, and in 1945, Fleming discovered the first antibiotic, penicillin. For this, they got the Nobel Prize as well. In 1962, Watson and Crick disclosed the structure of DNA and got the Nobel Prize. In the year of 1968, Hawley and Corana won the Nobel Prize for discovering genetic code, and in 1997, Pruesner also got the Nobel Prize for finding out about prions. Lastly, in 2002, Brenner and Havertz discovered the genetic regulation of organ development and cell death and won the Nobel Prize. There are many more inventions that are happening, which means microbiology is developing every single day. That's all for today. If you liked my video, then give it a big thumbs up and subscribe if you want to. Thank you so much.